In this video tutorial, I am going to discuss the units of enzyme activity. Do you know most of the substances in clinical laboratories like glucose, proteins, uric acids are measured in terms of weight like milligram and gram. But enzymes are measured in terms of their activity, not in terms of their weight. Now, there are two main units of enzyme activity used in biochemistry. These are international unit, abbreviated as IU, and Katal, abbreviated as CAT. So, the first question arises that why we measure enzyme differently in terms of activity and not in terms of weight. The answer is simple. In case of enzymes, what we measure is not how much the enzyme weighs, but how well it works, and that is activity. Let's try to understand this with the help of example. Suppose we have two samples of same enzyme, sample A and sample B. Both weights equal, let's say 10 gram. But sample A contains mutated and structurally altered enzyme, whereas sample B contains properly functional normal enzyme. So in this case, sample A has reduced enzyme activity, but sample B has normal high enzyme activity. So in this case, as you can see, the weight tells us nothing about the enzyme's ability to catalyze the reaction, right? That's why enzymes are measured in terms of their activity, not in terms of their weight. You can think of this like a water pump. A heavy pump could be broken, while a lighter pump could work perfectly. The weight of pump doesn't tell you how well it pumps water. Similarly, enzyme activity, not enzyme's weight, reflects its true functional capacity. So, new question arises, that is how to express enzyme activity. Historically, many different expressions were used, like King-Armstrong unit, Somogyi unit, Rateman-Frankel units, but currently the most commonly used expressions are international unit and Katal. Let's first understand international units or IU. In 1961, the Enzyme Commission of International Union of Biochemistry suggested this unit. Interestingly, they didn't give it any specific name. So different organizations called it different thing. Some called it as international unit, some other called it as just unit or standard unit or enzyme unit. But international unit becomes the most popular. So what is the definition of international unit? One international unit is the amount of enzyme that catalyzes the transformation of 1 micromole of substrate to product per minute under standard condition. One more time, one international unit is the amount of enzyme that catalyzes the transformation of 1 micromole of substrate to product per minute under standard condition. To better understand this definition, let me give you an example. If someone says that in this test tube, there is 30 international unit alkaline phosphatase is present, that means there is an amount of ALP enzyme present in this test tube which is converting 30 micromole of substrate to product in 1 minute under standard condition. Here, don't forget standard conditions because the enzyme activity is highly affected by various reaction conditions like temperature, pressure, pH, cofactors, etc. So activity must be measured under standard condition so that result of enzyme activity can be compared across all the different laboratories. One more thing, don't confuse this international unit with the international unit of biological activity. Both are completely different units. This international unit of biological activity is used for vitamins and hormones. It has its own separate definition, whereas this international unit is specific for enzyme activity only. Now, here is something important. Some books wrongly state that international unit is the SI unit. That's completely wrong, incorrect. In fact, there is a problem with international unit. These micromoles and minutes, which are used in the definition of international unit, are not a base unit in the SI system of measurement. However, scientists prefer using standardized SI units for consistency across all the fields. So in 1972, the same Enzyme Commission of International Union of Biochemistry recommended a new unit, that is Katal. So remember, Katal is the SI unit, not this international unit. Katal, abbreviated as CAT, is the actual SI unit for enzyme activity. Now let's look at the definition of Katal and compare it with the definition of international unit. One Katal is defined as the amount of enzyme that catalyzes the transformation of 1 mole of substrate to product per second under standard condition. Notice the difference with international unit. International unit uses micromoles per minute, whereas Katal uses moles per second. Both are measuring the same thing, enzyme activity, just with different units. Remember, international unit and Katal, both units are interconvertible. So relationship is 1 international unit is equal to 16.67 into 10 raised to minus 9 cat, whereas 1 cat is equal to 6 into 10 raised to 7 international unit. So by using this relationship, we can interconvert international unit and Katal. See, Katal is the official SI unit, and generally people all over the world preferably uses SI unit. However, in case of enzyme activity, most clinical laboratories still uses international unit over Katal. So why do most clinical laboratories still uses international unit? The answer is practicality. Katal values are very small for routine use. Let's understand this with an example. So for example, normal serum amylase activity is about 60 to 180 international unit per liter. Let's convert it to the Katal. So to convert it into Katal, we can utilize any of this relationship. It's simple mathematics. So to convert this 60 international unit to Katal, we can use this relationship. So we have to just multiply 60 with this 16.67 into 10 raised to minus 9. And so calculation will give us 0.000001 Katal. These are 5 zeros. And by similar calculation, this 180 international unit corresponds to 0.000003 Katal. Again 5 zeros. These are very small number and very inconvenient for daily reporting. That's why most clinical laboratories still uses international unit for daily reporting. So to wrap up, enzymes are measured by activity, not weight, because activity tells us how well the enzyme works. International units are still the most commonly used in clinical labs because they are most practical. Katal is the official SI unit, but it's less convenient for daily use. However, both are valid, both are interconvertible, and now you know why we use them. So that's all about this video. I hope that all the concepts are clear. If you still have any query or confusion, please write it down in the comment section below. Thank you. See you in the next video.